Okay, thank you. I'd like to start by thanking the organizers. I was invited to talk about regenerative medicine. I work at the Center for Regenerative Medicine in Barcelona, and our group is a part of Cyber BBN, so I thought I'd give you a broad idea of what we're doing and what we're doing in the context of Cyber BBN. So there are several groups inside Cyber BBN that tackle the three classical pillars of regenerative medicine: cells, biomaterials, and signals. We mostly work on, let me see, this works, yeah, on cells, but thanks to the collaboration with other groups of the consortium, we incorporate information from biomaterials and signals. The cells we work with are pluripotent stem cells. We truly believe that this is the future of regenerative medicine, basically because we're not thinking on delivering substances through cells, but truly regenerate organs or tissues. And these type of cells have the ability to be maintained, propagated, and expanded indefinitely in the lab, and then differentiate into any cell type. So they have the potential to generate cells that will be useful for many diseases. As early as 2009, we provided evidence that with pluripotent stem cells, we could generate them from patients. In this case, were patients with a severe problem in their ability to make blood. So we could correct that disease in the iPS cells, and then those were disease-free, differentiate blood progenitors that were as well disease-free. So there are this, at the time it looked like something that will be science fiction or medicine fiction. And there were many problems for the clinical implementation of these type of strategies. I will be very brief, but believe me that most of the problems have been solved during the past 9-10 years, such as the technical problems of how we make these cells, how we can use them or use a technology that will reach most of the patients. It's a very expensive technology. So all those problems have been solved, and I'll be glad to discuss with more detail if you're interested in that. And now we face the problems of safety, but as you probably know, the first clinical trial using this type of cells have been published earlier this year. So these cells have been already put into patients, and there were no safety concerns. More testing needs to be done, but we're already putting them into patients. And this is the main problem that we face currently, which is how to make the specific cell type that you want with the appropriate properties of maturation and ability to integrate into a person, into a host organ. And this is where most of the field is now, the trend in the field is focusing on this type of experiments. So for instance, we work on cardiomyocytes, and we can generate, we have very good protocols to make cardiomyocytes in the lab from iPS cells. They look like and behave like cardiomyocytes. You can obtain almost 100% of the cells in your 2D culture expressing markers and beating like cardiomyocytes. But the problem is these cells are very mature. They cannot be applied into human patients. This is also a problem for many other cell types. So what the field is doing, and for instance, we can take these cardiomyocytes, culture them in hydrogels with instructive biomaterials, and upon appropriate mechanical stimulation, in this case cyclic stretching, they align, the cardiomyocytes labeled here in red, they align and they start contracting in this type of hydrogels. I don't know if you can see them beating, but they are. Okay. If you want to have larger constructs, larger scaffolds, you can use porous scaffolds in perfusion bioreactors. They also promote maturation. If you cyclic stimulate, in this case using electrical stimulus, you can see how this is a non-stimulated construct that contracts. This is a large, around 1 centimeter per 2 or 3 millimeters thick. And this is the one that has been electrically stimulated, which has much more contractile force, and also has electrical activity, spontaneous electrical activity that can be measured in the surface of this construct, and it really looks like a human EKG, and is predictive of toxic effect of drugs, for instance, inducing arrhythmia that cannot be seen in any other animal or human-derived model so far. You can also take these constructs loaded with cells educated by electrical stimulation, implanting them into, in this case, this is a pig model of myocardial infarction, and they integrate quite well. They get revascularized, they get innervated, and they contribute to the functional amelioration in the pig. So this is where the trend in regenerative medicine using these cells is leading. We have same problem for heart muscle cells, for cardiomyocytes, as for any other cell type that you want to imagine. Many labs are working on solving these issues, but they are already being tested in humans, and there's much more coming in the very recent near future. So this will be an extraordinary opportunity to treat degenerative diseases in which the function of a specific cell type or tissue is lost, and you can regenerate this type of tissue. If the disease is caused by a genetic problem, since you can have the cells in the lab for extended periods of time, you can correct that disease and generate cells that are exactly the same as a patient, but they do not contain the genetic defect. While we were doing this type of experiments, the field in general realized that these cells were also a very good way to generate models of disease. Models of disease consistent on human cells that are otherwise unavailable from the patients. And in these models, we can study the pathogenic mechanisms leading to the disease. We can start asking questions about what the genetic susceptibility for risk or protection is in those patients, and also conduct all kinds of screenings. I'll give you just a few examples of this, but the basic strategy is to generate these cells from patients carrying a disease, and if there is any genetic component to the disease, it will be captured in the iPS cells. You differentiate the cell that is relevant for the disease. You don't correct the disease. In this case, you want the cells to be sick and to study the disease appearing in the plate. And you can see how some phenotypes appear in the lab that are reminiscent of what you see in the patients. This technology has been used to model many diseases, starting from the blood disease that I mentioned earlier, where you can see that the cells have the same phenotype that the cells from the patient. You can generate macrophages for Gaucher disease, for instance, and see that the macrophages also show the same problem that the patient's macrophages, or you can look at Sanfilippo, look at the neurons that you generate in the lab from these patients and see that the type of inclusions, abnormal inclusions that they have are very similar to the ones that you see in the patients. Or you can study hemophilia and see that the hepatocytes have the same problem in producing this factor that the patients have. So for monogenic diseases, it's very easy or almost immediate to have a phenotypical recapitulation in the dish, are very easy to study in the lab. But this technology is also useful for much more complex diseases, such as Parkinson's disease. You generate iPS cells from Parkinson's disease patients, and you see also the appearance of phenotypes, such as accumulation of alpha-synuclein, that also the patient neurons show, and neurodegeneration summarized here with these colored bars, so you see that all the patients have very great extents of neurodegeneration, and the controls don't. So with the advent of technologies such as CRISPR-Cas9, that allows us now very easily to genetically manipulate these cells, we can start investigating if a disease is of complex genetic origin and there are proposed or candidate variants that will increase the risk, we can start analyzing if this is true, because you can change that variant. For instance, in these patients, we have identified several hundreds of putative variants that confer risk or protection to the patients to suffer Parkinson's disease. And now we can directly edit, in this case is a rare variant that is predicted to confer protection to suffering Parkinson's disease, and has an effect in how the cell manages the Golgi functioning, the functioning of the Golgi apparatus. And we can edit that variant in our cells and prevent them from suffering Parkinson's disease. But these cells, again, can be differentiated into any cell type, so you're no longer constrained to using one cell type to analyze a disease, to model a disease, but you can generate complex co-culture systems with cells differentiated from the same patient or from controls and see what's the relative contribution of different cell types to the disease. Again, with this example of Parkinson's, you can generate co-cultures of neurons and astrocytes and see if the astrocytes contribute to the disease. And we see that when we culture neurons from controls with control astrocytes, they're fine. Neurons from controls on top of Parkinson's astrocytes from patients, they suffer and undergo neurodegeneration, highlighting that the astrocytes do have a role in suffering Parkinson's disease, and this is generated from cells from the patients. And again, CRISPR allows us to do things that we only dreamed about 5 years ago, such as, for instance, in this case, tagging the endogenous alpha-synuclein locus in astrocytes, so we can see where this alpha-synuclein is produced and where does it go. And it turns out the alpha-synuclein produced by astrocytes is directly uptaken by neurons and making neurons sick. So in summary, I hope I have convinced you at least that these new trends in regenerative medicine using this type of cells are not only giving rise to new strategies to replace cells, to do cell therapy and tissue engineering with this type of cells, but also generate really advanced, genuinely human models of disease. I'd like to thank the people from the lab and many collaborations that we have had with groups belonging to Cyber BBN, the funding for this work, and of course you for your attention, and I'll be happy to take any questions if you have. Or maybe later.
Yeah, nice work. Did you go that far as to check the mechanical cues in these systems? I mean, how the mechanical gradient is changing, especially when you study the neural astrocyte interaction?
Well, not in that specific context, we haven't. We have tested, of course, how mechanical cues control the maturation of cardiomyocytes, which are something, I mean, the low-hanging fruit. And in that context, they are critical, and we have explored them in some detail.
Hello, Angel. Thank you for your presentation.
Thank you.
I don't know. As you presented about the monogenic genetically diseases, and taking into account the possibilities of gene editing with the new CRISPR and these kind of tools, could we think about a potential cure for these diseases just by repairing this gene? and going forward with the transplantation of the cells afterwards? I mean, how far can we be of this application?
Well, that's a, we can spend hours talking about that, but in my opinion, what we are doing is not trying to cure the disease, okay? So we're trying to regenerate something, organ, tissue, cell, with the cleanest and healthiest cell that we can provide. So in that sense, we are targeting degenerative diseases, where the rest of the body is okay, is functioning, and only the dysfunction of a specific organ brings about the disease. You could think, of course, on using this type of technology to actually cure, but that will entail changing cells in the entire organism, probably going through the germline, ensuring that the next generation is healthy, cured from that disease, and that's entering into terrains that we are at the moment not exploring. Other groups are. I don't know if I answered your question. Perfect.
I have one question. How many years before the Nobel Prize for CRISPR-Cas9, you think? 2 years, 3 years?
I was expecting it for this year. Probably next year.
Probably. They will have it really soon. Thank you very much, Antoine.
Thank you.
So we go to the next speaker, which is Mark Ramis from Senolytics Therapeutics.